Hi everyone, in this tutorial we are going to discuss all about transgenic mice and how to make a transgenic mouse and what's the process that goes behind it, at least at a molecular biology level. So in front of me you have this diagram that we are going to dissect step by step and just see where it goes. But the rationale or the idea behind the transgenic mice is that when scientists are trying to study a specific disease or a phenotype that is particular to a disease, one of the best alternative instead of using human or higher mammals is to actually simulate the same functionality or physiological function inside another species, and we usually go for mice because of the fact that they've been extensively studied, and most of the time the mice that are used in the lab as well, they have quite a reasonable reproduction rate in terms of getting one generation after the other, and also they have quite a genome that has been extensively studied. But without boring you with all the details, what we're interested in first is understanding the process that goes behind it in terms of the techniques. One, you have micro injection. So what you do, you have the DNA, or in this case, it could also be a gene of interest. You need to inject it into the pronucleus of a fertilized mouse egg. And this is what happens here. You have a fertilized egg, and you are going to have your transgene, or the gene that you want to transfer. You inject it in this fertilized egg. Then, as soon as this process is successful, your next step should be implantation of this micro injected embryo now into a foster mother. So it won't be these two here, but you're going to find a different foster mother here in our step 2, where you are going to implant this diploid zygotic nucleus. And of course, once the foster mother has carried the nucleus, after a couple of days or weeks, you are going to have a couple of offsprings, and within those offspring, you are going to analyze those pups and subsequent generation for stable integration and as well as the expression of the transgene. But the way it happens, what you notice is that, let's say if this foster mother had four offsprings, the one with this chimeric and or mosaic type of patches here on the fur is going to be the hemizygous species that you are after. But of course, as soon as you notice this, you cannot just look at this phenotype and rely on it. You would want to actually test for the presence of this transgene, at least for this level, at a DNA level. So how can you do it? By performing a biopsy. So a biopsy is you just remove a portion of the tissue. For instance, you can just cut the tail and extract the DNA. Once you've extracted the DNA, you can run a Southern blot, which you know by now has to do with testing the DNA presence with hybridization, or you can simply run a PCR of the tail biopsy DNA. And by PCR, since you know the gene very well, you could design a couple of primers that are going to actually anneal to your complementary region. And if the DNA is present, you should see in that jar bands like this that are going to correspond to your DNA of interest. And once you have that in this mosaic hemizygote mouse, the next step is going to be now analyzing for the stable integration. So in other words, I have the DNA present, but is this integration stable? Something that is stable means that it will always be present and it won't be moved. All right. So how do we analyze now for stable integration? And in order for us to have the stable integration, what we'd want to do is actually breed this wild type, this mosaic mice actually with the wild type here. But before I go to that, actually, some of the information that we may also want to know is As we said, we transferred the embryo into the reproductive tract of the female mouse here, and some other facts as well. Usually, this is not an easy task, and only 2 to 15 of successfully injected embryos are actually transferred, and the pregnancy is visible for about after 2 weeks after, and these perhaps are weaned and analyzed at about 3 weeks of age. So that is also some relevant information that you may want to know as well. Now that we have that out of the way, let's go back and resume with our analysis of stable integration. For a quick recap, we are now at this stage here. You've done all that, you've checked after a couple of weeks, you actually saw the presence of this DNA, but we want to know, is this integration stable? And stable integration means that it will always be present, even if you pass it, even if you check from one generation to the next one, to the next one, to the next one. And the second thing that you also want at the end, you no longer want a hemizygote gene, but you want something that is going to be homozygous. All right, we're going to expand more on that very soon. So how do we do that? The mouse in which there was a successful integration, not stable yet, we just know it was integration, we refer to it as our founder, or the F0. So this one here is going to be our founder. It's our F0. Then we are going to now take this first founder, which is, like we say, mosaic or chimeric, because there's a mixture of things, as you see with the patch, and it's present in some of the tissues, but not all the somatic cells. So it is not integrated in the entire mouse yet. What we do, we try to create a F1 generation. And how do we obtain the F1 generation? Is by breeding our founder with a wild type. You see, this is coming out of Norway. We're breeding it with this one, and it's going to give us a F1 generation. So once you have your F1 generation, what you're going to see, if you test for the transgene again, 50% of your mouse or mice are going to be hemizygous. Of course, I've been talking about hemizygous, homozygous for quite some time now. Maybe you're wondering, what is this hemizygous? What is this homozygous? Let's just look straight into it as we approach the final concept phase of this explanation. So the next generation is all this F1 generation. As I said, once you have them, you analyze them again for stable integration. And if the germline of the founder is being transmitted in a stable manner, then what that will imply is that all the descendant of this mouse are member of this unique lineage. So everything that is coming out of this one now will carry a portion of that initial genes that we have. But the genotype now, in terms of genetics, of our founder are hemizygous rather than heterozygous. If you're confused, let's look at this picture here. Hemizygous, you see that this gene is only present in one allele. That's why it's hemizygous. Then, when you have this F1 generation now, you would see that it will become now, most of them are going to be heterozygous because we have a wild type. So one version is going to be what we're looking for, and the other version is going to be something different. So both allele are different. But at the end, we want to keep our inserted gene on both allele to be the same. And what we want to achieve now is going to be a homozygous, where both alleles are actually the same. And how do we get to achieve that is when now, after this F generation, we mate two of the F1 generation. If you mate two of the F1 generation, you are going to get now a new line here. And in this new line, what you will notice is that Again, you see here, these are the hemizygote 50%, so half of those. But in this one now, you're going to have 50% are going to be what? Hemizygous, and another 25% are going to be homozygous transgenic. That's what we're looking for. And another 25% is going to be homozygous wild type. And of course, what you're interested in is your homozygous transgenic, which is going to be this very dark mice here. And I'm going to repeat that again one more time. So once you reach this stage where you understand that I've got my F1 generation from breeding with the wild type, I'm moving from a hemizygous, from only one allele, to a heterozygous, because we've introduced a wild type here, which means that both allele, they carry a different version. They're not the same. And in order for us to get the same, we are going to crossbreed two heterozygous from the F1 generation. This one and this one, since they're 50%, what we're going to get now here is another 50%. This is just a Punnett square hemizygous, and a 25% homozygous wild type, and a 25% homozygous transgenic. And what we are after is this 25% homozygous here. So feel free to watch this over and over again, pause and repeat and all that. But at the end of the day, that's what you have. Once you have a successful transgenic mice, you notice that both alleles are going to be the same. As a result, you can carry on with your study and you have a successful transgenic mouse. Thank you.